My name is Blake Anson, and I work at Cellular Dynamics International, and we'll be hosting this exhibitor workshop. So the title of the workshop, as you can see here, is going to be Integrating Target-Based and Phenotypic Screening Strategies with Human Cardiomyocytes using IPSC Technology in clinical populations to identify new approaches for treating cardiac hypertrophy. So really what we're going to do here is we're going to talk about how the power of IPSC technology is really being realized by actually starting to incorporate information, both genetic and phenotypic information from populations of people, not just single individuals here and there. Okay, the talk will be broken down into two basic chunks. The first will be myself giving you an overview of IPSC technology and CDI's capabilities in utilizing this technology and getting it into your hands. And then we'll have Dr. Ulrich Breckel talk about how he's looking at taking genotypic information, combining that with phenotypic information derived from screens to look at new avenues and new markers of hypertrophy and potentially new therapeutic avenues. After all is said and done, if you don't walk away with anything else but these three messages, I hope you walk away with these three messages. And that's really that the IPSC technology allows you to bring relevant human biology into the dish. Secondly, it provides the appropriate endpoints. So you're talking about cellular biology endpoints, not just target-based endpoints. This is really the basis of phenotypic screening. And then last but not least, since you're not entering into these ideas or these screens with preconceived notions, you're able to uncover potential new pathways for drug discovery. And those are really the three main points, I think, to take home from a technology standpoint. Uli will have lots of other very interesting points as well. All right, so if we look at IPSC technology, as I've been saying, it really provides us with access to relevant human biology. Many models are already out there, and they all have their uses. We're well aware of those. You know, primary human cells, they're going to give you the biology, but there's going to be limited availability, unknown variability, unstable, and they will dedifferentiate in culture. Sorry, we're trying to adjust the volume here. Transformed cell lines, again, they'll give you an exquisite image or access to a single protein, but again, you're missing out on that whole cellular biology. And animal models, well, they're animal models. You'll have the biology, but it's not human biology. The induced pluripotent stem cell technology, on the other hand, has the potential to overcome all of these barriers, and I hope that that'll become clear as we throughout this talk. All right, so what is the power of IPSC technology? It's illustrated here on this crude graph. First of all, from stem cell technology, if you've got the right techniques, the right cooking, you can differentiate into any of the cell types of the body. The iPSC technology means that you can reprogram or get donor samples from just about anybody that you have access to. So we've got all the cells, all the genotypes, and then if we want to, we can go in and do genetic engineering, which we won't really talk about too much today, and manipulate any of these genes as well. So you're really able to get all of the biology and all of the really conceivable manipulations or investigations that you may want to do with that. All right, from our company, so is that promise that has really prompted the start of the company. Cellular Dynamics, we've been around since late 2004. We're headquartered in Madison, Wisconsin. We've got about 140 employees total. If you add up our experience handling human stem cells and their derived tissue cells, we've got about 650 to 700 years cumulative experience. It's a very complex legal field, so we've got over 800 patents either owned or licensed that allow us to do what we do. Also something that's very unique from our standpoint is that we've got a fully staffed support and application lab. Over a dozen scientists that are committed to generating protocols to show people how to use the cells and then provide help for people when they have difficulties using the cells. And as we'll talk about, our core competence is really our creation and culture of the stem cells from both normal and disease phenotypes, the engineering of those cells, so whether it's putting in markers, putting in cell type selection pathways, or just making various disease-specific mutations, we're very adept at that. We also develop new differentiation protocols. So you'll see we have a list of our off-the-shelf products, but we're always looking to increase that product portfolio and develop new differentiation protocols. All right. Then last but not least is really taking those laboratory exercises and taking them up to an industrial scale, right? So we can make not just 6-well Petri dishes, but billions and billions of cells. Right, our product portfolio is shown here. As I mentioned earlier, we have these standard off-the-shelf products, which are called iCell products. You can see they cross over the various germ layers. And we also have the Micelle products. And this is really what we're going to talk about for the majority of today. And the Micelle products is where we take in donor samples, do the reprogramming, and then make the terminally differentiated cells. And again, these donor samples can come from the wide array of apparently healthy and normal cells, or they can come from disease cohorts, or they can come from clinical trials, right? It's really the world is your oyster type of a thing. All right. Really the way we do this with the standard, with our manufacturing process is that we have a donor. These are for the iCells, so the scaling up of the iCell technology. We have our master cell bank of iPS cells. We'll peel off an aliquot of those, run them through the differentiation, purify and cryopreserve them. We've got this platform worked out such that we can generate, on average, billions of the various cell types per day. We've really taken this out of the hood and into the industrial setting. All right, the scale out aspect of it, though, is to be able to do this across hundreds and thousands of donors. This, as I alluded to earlier, is the Micelle. And a couple of the examples of this, of the way that we're building out this disease and diversity panel, is, as we'll talk about later, this cohort of patient samples that all had left ventricular hypertrophy. This will be the basis of Uli's talk. We've also got other aspects going on, or other endeavors going on, where we've got an award from CIRM, California Institute of Regenerative Medicine. We're going to be making reprogramming iPSCs from 3,000 donors, and doing three clones from each donor, so 9,000 total samples. They've got smaller endeavors looking at neuronal, hepatic, and other cardiovascular endpoints. So we're really covering the wide gamut. What we're going to focus on today, though, is I'm going to talk a little bit about how iCells are being used in disease modeling and screening. And then Uli will talk about this left ventricular hypertrophy scale up of the GWAS samples that he has. All right, so iCell and Micelle tissue cells. Well, the first thing is, what's the general description of the cells? And that is, any of the iCells that you would get from CDI are going to be highly pure, cryopreserved, and ready to use. It's a plate-based unit size, so one vial will equal a plate. And you can order the unit sizes in virtually unlimited volumes. All right, they'll all show the expected human biology, and it really is the industry standard at this point. We've got a broad platform utility. We've got more customer publications than all the other commercial providers combined, and it's utilized by over 90% of the top pharmaceutical companies. So it's not just a science fair experiment anymore. It's becoming a mainstream of drug development. All right, the Micelle, here again, is where we bring in donor samples. These are reprogrammed using an episomal or footprint-free method, meaning there's no integration of those reprogramming factors into the genome. So you've got a clean genome. It's optimized to work on small sample volumes, really just a few mils of blood. And then also the reprogramming conditions are all feeder-free and defined, so you're able to trace back everything that's ever touched these cells. The genetic engineering, again, we have a variety of ways of doing gene editing for a variety of endpoints, and just suffice it to say that we are well versed in virtually all of the technologies that are out there. Then last but not least, we've got the differentiation. So either into the catalog cell types, the iCells, or into custom products that we can enter into discussions with. All right, none of this is any good unless we can help you use the cells as well. And so we've got partnerships and co-marketing efforts with a variety of endpoint users that allow you to interrogate a variety of the ending procedures. So you not only get the cells, but you get a head start or a heads up on how you can use these cells. You're not having to reinvent the wheel. All right, we also then, for the eye cells, we provide a very rigorous characterization of the cells. And I've got an example up here for the cardiomyocytes. This is accumulation of several years of work and probably around a dozen or so publications simply on characterization. But suffice it to say that the gene expression is stable and what you'd expect for cardiac cells, human cardiac cells. The protein expression, it's the expected protein complement, and it allows or enables the function. Metabolism is going to be omnivorous, so it's both fatty acid oxidation as well as glycolytic, just like mature cardiomyocytes are. The biochemistry is all of there, so your kinase inhibitors, your secondary pathways, calcium handling. Excitation-contraction coupling, which moves the electrical activity of the cardiomyocyte to the actual movement, is all there. So really, as an example of the eye cells, we've got a fully characterized cell type, and it shows that it's the expected biology that one would expect from human cells. And really what this does is this then accelerates your research, because it gives you the relevant phenotypes, the appropriate targets, and an easier translation to the native biology, to the in vivo biology. Right. Now, the Micelle samples that we come in, we can't characterize them to this extent because this is one cell line versus the thousands of cell lines, but they're all made through the same process. So the ending cell that you have come out is all subjected to the same treatment, and you ultimately end up with the cell that still provides that relevant phenotype. All right, if we're going to give you, I think, 3 examples here of just using the cells in in disease modeling and screening. And it's important with this access to this tissue now, you can approach it from disease models from an induced aspect or an innate disease model. So environmental versus genetic or genomic conditions. 2 panels here from a paper published by GSK looking at Alzheimer's, so looking at specifically at A-beta toxicity in Alzheimer's. The simple phenotype here is simply neuronal viability, and the experiment was to simply expose cells to the A-beta peptide to induce the toxicity, and then screen for compounds that are going to rescue that viability. And what they found here is you've got several compounds that gave them positive hits. They were then able to follow up with a variety of techniques such as siRNA for knockdown. Right. This work was published a couple of years ago, and as far as I know, is still being worked on within GSK. All right, if we look at another example of induced disease states in eye cell cardiomyocytes, would be looking at cardiac hypertrophy. Right, the native phenotype here, normal-sized heart becomes larger with enlarged muscular walls. Some of the cellular hallmarks of hypertrophy are going to be increases in cell size, enhanced protein synthesis, sarcomeric organization, and a reactivation of the fetal gene program. This really is the main readout that we use in our hypertrophy assays. That is the upregulation of BNP. But we've taken this, initially we took this phenomenology and sought to determine if we could recapitulate that in eye cell cardiomyocytes. And we can. So if you induce with endothelin, you can see a dose response increase in that fetal gene expression. So here BNP. Cytoskeletal rearrangements, here a change from F to G actin, as well as an increase in cell size. So we've got the appropriate phenomenology. The next step was, can we actually make this into a rigorous or a robust screen? And the answer there is yes. So again, we've got the endothelin induction. Here we're seeing what I just showed you earlier, the increase in BNP production measured through high content analysis. We can also measure that through qPCR, so looking at message, looking at flow cytometry, or looking at ELISA. And again, we've got the standard, similar EC50s across across each of those readouts. So we've got a nice, robust assay. The Z factor for the assay is quite high as well. So we're up here around, we can't even, 0.8 to 0.6. Sorry, my eyes are getting old, as am I. We've got a highly robust Z factor, so that's suitable for screening. And it's also got a responsive phenotype, meaning you can induce the BNP phenotype and then screen it away with a variety of small molecules. Again, doing this in a phenotypic manner, you're not having any ideas about the target. All right, so that's an induced phenotype. If you look at an innate disease model, that's this one right here, where you look at the R403Q mutation in the myosin heavy chain gene. This is responsible for about 50% of the familial hypertrophic cardiomyopathy cases. We brought this sample into the Micelle program, reprogrammed the sample, did the differentiation, and we make fully functional cardiomyocytes, except for the fact that the baseline gene readout of the diseased cardiomyocytes is very similar to that of the induced normal cardiomyocytes. So we've got a propensity, or these cells have a propensity for that hypertrophic phenotype right off the get-go. Right? You can see here from expression levels where you've got virtually no expression of BNP in the isol cardiomyocytes, you've got a lot of BNP expression here at the baseline level for the mutation, for the mutated cardiomyocytes, or the clinical cardiomyocytes. Similar to the wild type, we can further induce that phenotype with endothelin, and it can be competed away, in this case with verapamil. So again, we've got a screenable system, but now it's from a disease phenotype or a donor sample. All right, and it really just provides another model or example of that innate disease models. All right, so I think this is my summary slide here. So we've discussed the modeling and screening with either induced models or SNP-based innate models, right? Single nucleotide change, amino acid change, gives you the disease phenotype, right? But what about the power that I've been talking about of iPSC technology, right? How can we bring populations into this arena? And one answer is, as we'll talk about, is through this NHLBI study that Uli is in charge of. It's basically here, we're 250 samples, patient samples from the hypergen cohort. So these are individuals that are suffering from left ventricular hypertrophy. 250 of them, deemed interesting by the geneticists and the clinicians, were chosen for reprogramming and iPSC analysis. Right? So CDI derived the iPS cells, cardiomyocytes. You can see here, if we look across several different lines, we're able to provide consistent cardiomyocyte populations across different lines. So all of these here are greater than 90% purity, and we see a very high percentage of ventricular cells across lots. These are then provided to Uli for him to do his hypertrophy and molecular analyses. And really the take-home message from this slide, because I'm not going to steal any thunder from Uli's results, are that these types of population studies are ongoing, they're feasible, and they're generating valuable data. So with that, I'd like to turn it over to Dr. Bracco and have you come on up.